You know, in the world of microbiology, some of the most interesting culprits are the ones hiding in plain sight. Today, we're unmasking one of the great mimics, Aerococcus viridans. Now, this is a pathogen with a pretty serious clinical punch, but its real danger? It's the uncanny ability to be mistaken for something else entirely. Just so you know, this entire explainer was synthesized using Gemini Deep Search, and the visuals you'll see are from Microbe Canvas. If you want to do a full dive into all the sources, just check out the linked notebook. So, let me ask you this. Is this organism a lobster killer, or is it a human pathogen? Well, what if I told you the answer is both? In the marine world, it's actually notorious. It causes something called Gaffkemia, which is a fatal septicemia in lobsters. But you bring it into the clinical world, and it becomes this often overlooked cause of some really serious human infections. And that dual identity is what makes it so fascinating. And this right here, this is the core of the problem. The greatest strength of Aerococcus viridans is its incredible ability to mimic other bugs. In the lab, it can look just like Streptococci, or Staphylococci, or even Enterococci. And this mimicry, well, it leads to misidentification, which can dangerously underestimate its clinical impact and, of course, delay the right treatment. So, to really get a handle on this organism, we're going to look at it from all angles. We'll start with why it's so often underestimated. Then, we'll head into the lab to unmask its disguise. After that, we'll look at the real-world clinical damage it can cause, and then finally, we'll map out the treatment playbook and how to keep it in check. All right, let's kick things off with Section 1, the underestimated pathogen. Where exactly does Aerococcus viridans fit into the grand scheme of the microbial world? You might hear about it causing Gaffkemia in lobsters and think, okay, that's a neat piece of trivia. But it's actually more than that. It tells us something really important. Aerococcus viridans is an environmental organism. It thrives outside the human body, and that gives it this unique and often overlooked zoonotic potential. Now, if you want to understand why it causes so much confusion, you just have to look at its family tree. Taxonomically, it's in the order Lactobacillales. That makes it a really close cousin to some very well-known characters like Streptococcus and Enterococcus. This close genetic relationship is the absolute root of the diagnostic mimicry we see on the lab bench. So let's move on to Section 2. This is where the rubber meets the road. We're going to do a step-by-step guide to unmasking this mimic right there in the laboratory. Your very first clue, and honestly, maybe the most important one, is on the gram stain. Okay, it's a gram-positive coccus, we get that. But the key is the arrangement. You need to be looking for tetrads, those little groups of 4. Yeah, you might see some pairs or small clusters, but the presence of tetrads should be a big flashing light that makes you think beyond your typical streptococci in chains. Then you get to the culture plate, and the mimicry just continues. It grows perfectly fine on sheep blood agar, and it produces that classic alpha hemolysis, that greening effect. Macroscopically, honestly, the colonies are often completely indistinguishable from viridans group Streptococci. But here's a key negative to remember. It does not grow on MacConkey agar. Now this slide, this really lays out the biochemical puzzle perfectly. The critical clue is the combination of tests. Aerococcus viridans is typically PYR positive. That's the test for the pyrrolidonyl aminopeptidase enzyme. But here's the kicker. What really sets it apart from other clinically relevant Aerococcus species in Enterococci is that it is LAP negative. It lacks leucine aminopeptidase. This PYR positive LAP negative signature, that is the unique key that unlocks its identity. So let's put it all together. The modern diagnostic workflow is a pretty clear 3-step process. Your suspicion starts with those tetrads on the gram stain. Then you get your differentiation from those key biochemicals. But for definitive, rock-solid confirmation, especially if the results are a little ambiguous, your gold standards are going to be MALDI-TOF mass spectrometry and 16S RNA sequencing. Okay, so now that we know how to find it, let's talk about why it's so critical that we do find it. Section 3, clinical impact. This organism is way more than just a potential blood culture contaminant. It can cause truly life-threatening infections. Let's talk about the big one, infective endocarditis. This is, without a doubt, its most severe manifestation. It usually shows up as a subacute infection, often hitting the native valves in patients who already have pre-existing heart defects. And the real danger here is that its slow, smoldering presentation can delay the diagnosis, which can lead to severe complications like embolic strokes and even heart failure. So how do you know when to escalate the workup? When do you really start to worry? Well, this is where a clinical tool called the de novo score becomes absolutely critical. It's a scoring system that helps assess risk factors for endocarditis. For a patient with Aerococcus viridans bacteremia, a de novo score of 3 or higher is a massive red flag. It's highly sensitive for endocarditis and should immediately trigger a transesophageal echocardiogram, or TEE. But it's not just endocarditis. Aerococcus viridans has a few other tricks up its sleeve. It can cause bacteremia, especially when it's related to central lines. It can cause urinary tract infections, particularly in elderly males. And it can cause these deep-seated bone and joint infections like spondylodicitis. And while it's pretty rare, it has even been found in cases of meningitis. All right, let's move on to Section 4, the treatment playbook. We're going to compare the Dutch SWAB guidelines with the international recommendations to really build a solid therapeutic strategy. Now, treating this organism presents a unique challenge, especially for the lab. As of 2025, EUCAST, that's the European Committee on Antimicrobial Susceptibility Testing, they don't have specific clinical breakpoints for Aerococcus viridans. So what do? The practical, recommended strategy is to use the breakpoints established for viridans group Streptococci as a kind of surrogate to interpret your susceptibility results. So, following those principles, the Dutch SWAB guidelines are very clear and straightforward. For native valve endocarditis, the go-to agent is high-dose benzyl penicillin, that's penicillin G, for a full 4 weeks. Ceftriaxone is an excellent alternative, especially for patients who might have a non-anaphylactic penicillin allergy. Now, the international AHA/IDSA guidelines, they add a little more nuance to the picture. They stratify treatment based on the specific minimum inhibitory concentration, or MIC, which is basically just the lowest concentration of an antibiotic that stops the bacteria from growing. For highly susceptible strains with a low MIC, treatment is pretty much the same as the Dutch guidelines. But for those relatively resistant strains, they recommend adding gentamicin for the first 2 weeks to get a synergistic killing effect. The crucial point when you're treating kids is that you have to throw flat dosing right out the window. It is all about strict weight-based calculations. Daily doses for drugs like penicillin G and ceftriaxone are prescribed in units or milligrams per kilogram of body weight per day. You have to do this to ensure both safety and efficacy. And that brings us to our final section, infection prevention and control. How do we manage this environmental organism once it's inside the walls of a healthcare facility? Well, the good news here is that Aerococcus viridans is not considered a highly transmissible pathogen. You're not dealing with some superbug that's spreading all over the hospital. For the vast majority of cases, standard precautions, which really just means diligent hand hygiene, are completely sufficient. You'd only need to think about contact precautions in the rare case of a major uncontained draining wound. Okay, let's boil it all down. If you remember nothing else from this explainer, please remember these 3 things. First, on the gram stain, when you see gram-positive cocci, think tetrads. Second, in the lab, burn that unique PYR-positive, LAP-negative biochemical signature into your memory. And third, clinically, treat its endocarditis aggressively, just like you would for a viridans group Streptococcus. So, the final question I have for you is this. How will this change the way you approach your next catalase-negative, gram-positive coccus isolate? For more deep dives just like this one, make sure you subscribe to the channel. And please, share your own experiences with identifying Aerococcus species down in the comments below. We would absolutely love to hear from you.